All right. Am I audible and my slides are visible to all of you? All right. Thank you. So today, this is lecture number 6 and in our 5-day course. So very good morning to all of you. Yesterday, we talked about multiomics. In fact, from day one, we are trying to learn this completely new paradigm of treating cancer. And somebody asked me yesterday about this paradigm being useful even for preventing cancer. So I answered that yes, it is possible that we can have precision and personalized approach for preventing cancer, but still this whole concept is catching up because concept alone would not be enough unless we have the right tools to implement it. And that's what I'm going to address today. So even if you think something is right, but if you can't do it, if you can't show it, it's not going to be taking off. So fortunately, I would say that multi-omics is getting a lot of traction from the development in the technologies that are helping us to generate the data, to analyze the data, and come up with this paradigm of multi-omics application in precision and personalized medicine, especially for heterogeneous disease, multifactorial diseases like cancer. So I gave you the examples in my first lecture that how the self-driving cars were progressing. This was a fancy concept, a science fiction novel concept, but as soon as the technology was mature enough for collecting the data using the sensors, using the cameras, and they were able to capture 360-degree view of the street ahead of the car, this became a possibility, along with the computational power to analyze this data in the real time. So both data collection and data analysis progressed tremendously, and that made these self-driving cars possible. And now it's a reality. They are running on the streets of America, and soon you might see it in Jabalpur, in Aligarh, or somewhere in India as well. So it's not something that we should not think about because it is very difficult to do, but we should be able to even improve the techniques. So let's go into specific techniques that have been employed in multi-omics approach. As you can guess, that there are different layers of multi-omics. So there would be layers of techniques as well. Some of the techniques are common, but some of the techniques are distinctly different. So just a recap that we have got several layers. Here we have put just 4 layers: genomics, epigenetics, transcriptomics, proteomics, which are the main, in fact, the central dogma of DNA, RNA, protein. That is something that we focus on. And nucleic acid genomics is taken care of by the techniques that are similar. They are employable for both DNA and RNA. So genomics and transcriptomics will be taken care of by one set of techniques. Then protein is different, so we'll have different techniques of proteomics. And metabolites are also different, so there will also be different techniques for metabolomics. But due to some similarity between the two, they are commonly used, like you'll see in subsequent slides, that there are some common techniques utilized for doing proteomics and metabolomics. So with the proteomics, the techniques mostly mass spectrometry. And in case of genomics, we are not talking about next generation sequencing, but there was a time when microarrays were very popular. And I myself have done a lot of work on microarrays in the US. So I know it was a lot of data that we used to generate, and it made a lot of sense, but technology took over, and next generation sequencing is now the standard these days. As I said, transcriptomics would be also employing the same technologies, next generation sequencing and microarrays, because the nature of the two is the same. Both of them are nucleic acids, so they will be using the similar techniques. For epigenomics, we also have sequencing, but there's some conversion that goes before uh, we do the sequencing, which is called bisulfite sequencing to take care of the methyl groups and the chemical groups that are modifying the DNA. And there's another technique called ChIP-seq, which is chromatin immunoprecipitation assay. So first we immunoprecipitate the chromatin using the antibodies, and then once we are able to do that, we do the sequencing. But this is not going to be for the entire genome. It is going to be specific for certain parts of the genome. So this is known as ChIP-seq. For metabolomics, we have got LCMS. So as you can see, metabolomics and proteomics. Can you see my cursor? Is my cursor visible to you guys or no? Okay. So as you can see, metabolomics is employing LCMS, liquid chromatography, mass spectrometry, and proteomics is also employing mass spectrometry. And here it is gas chromatography, mass spectrometry for some volatile metabolites that cannot be detected in liquid state, but in the gaseous state. So GCMS is going to be important for metabolomics. Now, again, I tried to emphasize on these things because we are doing multiomics because 90% of the cancers, they cannot be explained by mutations alone. I think yesterday talk and the other talk that you must have heard that this mutation in that cancer, that mutation in this cancer, but 90% of the cancers cannot be explained because this is not the work of one mutation or one DNA, rather it's a work of many molecules together. And subsequently, 75% of the cancer drugs can be ineffective in 75% of patients. So mostly you would hear that this drug did not work because it's mostly hidden trial. So again, we have to emphasize the importance of using these techniques in generating robust data that can be analyzed for precision and personalized medicine. So this is a broad overview of how do we do multiomics based on the current technology that we have. So I think yesterday we saw how we did the sample collection from the surgery. So when you take out the sample from the surgery from a patient or say, for example, blood, it's not a homogeneous mixture of cells. Rather, it's a heterogeneous population of cells as denoted here by different color shades that the cells are different. This is all heterogeneous. This is known as intra-tumor heterogeneity. If we are talking about the tumor, solid tumors, it's intra-tumor heterogeneity. So this is a starting material. Now, this can be processed as a whole, which we call as bulk cell analysis. And this is the mostly employed technique because we bring the tissue, we crush it, and we take out the DNA, RNA, protein. But this is not from a single type of cell, rather it is from multiple type of cells. And we call it bulk cell analysis, and the expected data is something that is a representation of all the heterogeneous cells present in that sample. Technologies evolved, people started questioning that and I also told you yesterday about laser capture microdissection, where you can pick up these individual cells by looking into the microscope. And I have done that myself. It's a very tedious work, spending hours and hours in the dark room, looking at the microscope and isolating single cells. So that technique is also becoming obsolete because of the problems in doing it. So you can pick up these cells manually and then use them for extracting the DNA, RNA, and protein, and then do the work. But now with the help of single cell analysis, this is possible to sort them out. So you can see the different cell types and characteristics, they can be sorted out in different populations, and then you can use those populations for carrying out your analysis. So this will give you a better picture of the DNA expressed in these dark colored circles versus light colored versus the lightest color. So you'll have a fair view. It's like a high resolution picture, right? So if you have got a high resolution picture, you are able to see the picture even to a level of the pixels. And then there's something in between which is called spatial analysis. You don't isolate these cells separately, but you are able to say that this particular DNA or RNA or protein is expressed in a cell which is located in a particular location. For example, here I can say that the bottom right corner of the cells have got a particular type of DNA, RNA, or protein modification. This is known as spatial analysis, which means we are able to put them in context that where is this kind of cells located. Spatial uh, details are important because we have to know where the cells are located, which are causing the problem, whether they are on the surface or they are in the interior of the tumor. If they are in the interior of the tumor, the problem will be different. And if they are in the surface, we can target them easily and there will be a different strategy for killing them. So yesterday we got through the sample collections that we collected sample from tissues or animals, and then we can make organoids or cell lines. So these are the starting point for the subsequent technologies. We have to have the material in the form of cells, whether it is extracted from the patient tissue or from the patient's blood, or it is taken from the animals from which we are studying, or we are developing the organoids, or we are talking about cell lines. So these cells are the starting material. And my aim would be to introduce you to as many techniques that are relevant so that when you end this course, at least you're familiar with what is available in this area in the market. If you go somewhere for your interviews, so you're not ignorant about these techniques, rather you have heard about it. But it will be very difficult for me to explain every technique in detail because I can tell you that each and every technique can be a lecture in itself. So I will be skimming the surface, but telling you what is the latest and what is not working anymore so that you have a fair overview of using these techniques for your own research. So genomics techniques, here is a little bit of evolution of how it happened. I think you might be familiar with the discovery of DNA and the double helix structure. And then the Catamulus invented this PCR machine. So these are all laying up the groundwork for what we see today. So there was another breakthrough of the next generation sequencing in 2009. So you can see that the whole genome, which was sequenced in 2001, the next generation did not exist, next generation sequencing. It was done through classical Sanger sequencing. I'll tell you a little bit about that in a bit. So it was a humongous task done by several countries together to have the first draft of the Human Genome Project. And recently, this 2012 initiation of whole genome sequencing has started in UK in 2012, so almost years. And India also recently got a project where they are mapping 1,000 genomes from the Indian population. And so this is also now in the public domain. As you can see here, while we are progressing here, we are increasing the amount of data that we generate. Right, so the amount of genomic data that is created due to these technologies is huge. But analysis will definitely become difficult as well. So Sanger sequencing was easy to analyze. I remember even looking at the films, we used to do it manually. ATGC, ATGC, CGTA. So we were able to do the sequencing by reading the autographs manually. But with the increase in the technology, this analysis is becoming more and more difficult. That is another challenge. So I'll touch base upon all these in my subsequent slides. Starting from Sanger sequencing, this is the most basic type of knowing the sequence of the nucleotides in a fragment of DNA. So the simple steps are we need to first denature it, we need to separate the two strands. Then by using the PCR, we make multiple copies of it. We attach a primer so that we can start the color-coded or the radioactively coded nucleotides. So these are the 4 types of nucleotides, which are either radioactively labeled, so we can see them when we develop the film, or it could be color-coded, that can be done through the automated sequencer. Then these are grown through the polymerase reaction, and then we again denature, and then we subject these new chains to the sequencing. And as you can see that each one of these chains is going to be one base different. So when you run them on the gel, they are all coming at the difference of one base. And this is how we read the sequence. So the sequence would be read as G, then again G, then it's A, then it's C, then T, then again G. So I remember using these autoradiographs. These were actually labeled and reading them from the sequence before the automated DNA sequences were available to us. So but it's not very old. It's like 20 years ago. The technology has transformed very fast and from this manual to the current level. Then we had the microarrays. In microarrays, it was based on hybridization. So on a single chip, which was a very small, 2 centimeter by 2 centimeter, we were able to put the millions of probes. Probes were just the DNA sequences like oligos. So if you have a sample from a healthy cell or a pathological cell, you can just put them together and they are coded. And this coding would be seen in the form of the color. So if there is no color, that means it's not present in the cells. But if it is red, it means that this gene is expressed in the disease cells. And if it is green colored, it would mean that it is present only in the healthy cells. And if it is yellow, it means it is present in both cells. This is how we can see the presence of a particular gene in a particular type of cells. And this was very quantitative as well. So by the intensity of the color, we could tell that what is the level of expression of this gene or what are the copy number of this DNA in a particular type of cell. So this used to be, but there was a lot of background noise here when we used to do the hybridization. So this was replaced by next generation sequencing. The next generation sequencing is actually the extension of the Sanger sequencing, but at a higher level. In Sanger sequencing, you are able to do only 200 to 250 base pairs, and that's also for a small portion of the gene. If you have to do a gene of 1 kilobases, you need to do at least 4 to 5 sequencing. And that is also possible only one time. In case of next generation sequencing, we say depth. So we are able to do multiple sequencing of the same fragment of the DNA. And by doing this, we are increasing the confidence as well as the statistical prowess of the sequencing data. Because if you see here, if this data is done once, it might be wrong. There could be some technical errors here. There could be errors because of the polymerase. But if the same thing is done 1,000 times or 100 times or 500 times, it makes more sense. So that's what next generation sequencing does. So it sequences the same portion of the DNA multiple times. And also it can sequence several thousands of these fragments by using the different chambers, which are small chambers or small bubbles in which these are going on in parallel. So this is also known as the next generation sequencing is also known as massively parallel sequencing, which means there are thousands of sequencing reactions going on in parallel in different drop-like structures. So here's an evolution of sequencing technologies. Sequencing technologies have also evolved a lot, and it is continuously evolving. So if somebody from the engineering background or somebody from the technical background, they want to focus on the technology, this itself is a big thing to do. So like Sanger sequencing, it started somewhere in the 70s, and then it was spiral sequencing. Then there was iron tolerant sequencing. Then Illumina came in the market. Still the market leader is Illumina. It is having the best setup for doing the sequencing. But these all were limited because they were not able to sequence more than 500 base pairs at a time. And you all know that the human genome is 3 billion bases. So, and when you are seeing things in small fragments, then you're not seeing the complete picture. And it was very difficult to put them together. It's like if you have a ball of wool and you cut it into small pieces, and then you are asked to put them back together, you can understand how difficult will it be. You'll also feel like if these fragments were bigger, the task would be at least a bit easier. So with that purpose, the long read sequencing started. So long read sequences was done by PacBio and Nanoport. These are the technologies, these are the companies who developed the technologies. And they are now able to go into kilobases or megabases. But there's still no technology which can do the entire genome in one go. It's impossible as of now, and not even near to that, because if you see one kilobase or one megabase, still we are far behind in doing the whole genome sequencing. Right. So another important thing is when the human genome came out in 2001, it was a project done by the entire US government, along with many other governments of the world. So it costed millions of dollars. It was written here, it's $100 million for the sequence to be done in 2001. There's an interesting story about a private company, Craig Venter jumped in and they did it even cheaper way, but that's a separate story. But as you can see, with the passage of time, the sequencing economics have gone down. So it's now less than $100,000, and it's even some companies in China, they are claiming to do $100 genome. What it means for multiomics, that yes, now patients are routinely getting sequenced for their disease, and it's easy for the clinicians to order these, and it's easier for the insurance companies to afford it. So that's why now it's coming into the mainstream. In fact, in Iceland, they have sequenced their entire population. So, like at the birth, you get your Aadhaar card or some kind of an identity card. They get their complete sequence. This is your sequence. If you ever need it, this is going to be available. And this is a kind of summary for the various next generation sequencing technologies. So this is pretty comprehensive. You start with the common thing is the library preparation, where you start with a sample genome. You cut down this entire genome to small pieces and then put your own codes there so that you can identify it later on. This is known as library preparation. And then each one of them starts to go under sequencing in a separate drop-like chamber. And then it can be either long reads, which means it's more than 500 base pairs, and it can be up to megabases. And the short reads, which is from 50 to 500 bases. And then long reads, as I told you, it's PacBio and Nanoport. These are the only two companies which are providing this service and equipment. So they are the leaders in their field. But in short reads, there's a lot of competition as well. But still, the Illumina is the leader as of now. There are other companies like Solid and Ion Torrent 454, Pyro Sequencing. They all differed in how they were doing the PCR and the subsequent sequencing. So they are doing emulsion PCR, which means they made the small droplets in which they did the PCR, and nanoball PCR and rich PCR. So these short reads are still there. It's very much used, and it's still is being utilized in clinic mostly. But Illumina platforms are leading the game. So we have got this different variants of this next generation sequencing as well. I'll tell you about it in detail in a minute. But mostly you categorize them as, this is the DNA level, genomics, whole genome sequencing, and whole exome sequencing. Whole genome means entire 3 billion bases. Exome means the portion of that genome which is expressed, which is roughly 25,000 genes. So it's about 1% of the entire genome that is expressed. So here the scope is much lesser, but you get more information, and because even if you sequence the whole genome, you don't know what is it doing. It's like dark matter, which has got no known function. So this data come out in the form of point mutations. You can find out small insertions or deletions. You can find out copy number variation, whether there's a increase in the copy number. Usually we have got 2 copies, but it could be 3, it could be 1.5, it could be 1.8, it could be 3.2. Structural variation, there could be a loss of an arm, like in case of colorectal cancer. Loss of 18q chromosome is a very significant step. And then you can also use it for lineage identification. All of us know about the DNA fingerprinting. That was used for kind of knowing where is the DNA coming from, and it is useful in forensics, paternity tests, and other things. Then we have got the RNA, and this is known as RNA sequencing, RNA-seq. I'll be dealing about all these things in a bit of detail in a few minutes. So the data that comes out from transcriptomics, that is in the form of differential expression, which means you get to know which gene is expressed more. Expressed more means having more numbers of mRNA, more copies of mRNA, because that translates directly into more protein, and which could translate into more function. The differential expression is some of the genes that are upregulated or downregulated in a particular disease condition, we get to know through differential expression. Then there are gene fusions. Gene fusion is there is a classical example in lung adenocarcinoma, NTRK fusions, where the one gene fuses with the other gene due to the chromosomal rearrangements. And this causes cancer. It's also common in blood cancers, especially in pediatric cancers. This gene fusion is quite common. We get to know through the transcriptomics. Alternative splicing is another level of complexity, because within the RNA, there are exons and introns. So as all of you would know, that exons and introns are patches on the RNA, the pre-mRNA, I would say. And before getting into the translation machinery, the introns are chopped off and exons are fused together. Now, this can happen in different combinations. I haven't put any slides here, but this is a separate topic to talk about. But alternative splicing generates different types of RNA depending on the need, which means we can have different proteins depending on the need. And RNA editing is there as well, the addition of the poly A tail or addition of a cap at the 5' end. These are all known through the transcriptomics. Then we also have epigenomics, where the addition of methyl groups can be studied. I just told you about ChIP-seq or ATAC-seq. They're going to tell us about the methylation patterns, if there is any modifications in the histone proteins, or if there's any modification on the transcription factor binding, because transcription factors are the master regulators. They are like the people who are orchestrating the entire gene expression. So it's important to know about the binding of these factors, which ultimately results in differential expression or the functional aspects of the RNA. This is something I've just put it here, but I'll not be telling you in detail about this. This is a microbiome. I think I touched upon it a little bit yesterday, the day before. So we can also use the techniques to sequence the microbes that are living inside our body by using the 16S ribosomal RNA sequencing, or the shotgun approach or targeted panel. And by this we can know what bacteria is living within us, what fungus and what microbiome as in general is living within us, or we can have the antimicrobial resistance gene profiling. So in case of cancer, there's a strong evidence that is coming up that microbiome is disturbed. So we can know by knowing the change in the bacteria or fungi living inside our body, that what is the status of the cancer, or how is it going to affect the progression of the cancer. But this is not the topic of the lecture or the course, so I'm not going to do it in detail. And all these information that needs to feed into the effects, like what is the effect of this mutation. So we do the EQTL analysis, which is the quantitative trait loci, that what is the effect of this mutation on a particular phenotype, or we can do network and pathway analysis, because as I told you in the beginning, these genes, they crosstalk, they know how to find alternate ways of survival. So we need to understand them from the network perspective, that let's see how these genes are connected so that we can make sense out of their differential expression. If one gene is upregulated, probably the second one is downregulated, and both of them are coordinating with each other. This has to be known through the network and pathway analysis. And integrative analysis is the same when we are combining multiomics and providing a full picture of the cell or the tumor. All right, so we go into a little bit of detail about the whole genome sequencing. So is everybody following here? Let me see if there are any questions. Because I'm not able to access, okay, no, there's no questions here. So you can speak out if you want to ask questions, because I'm on a separate tab and I'm able to see that. All right, so whole genome sequencing, as we just talked about, this is about sequencing the entire 3 billion bases. Now, why would we do it? Because sometimes you want to know the whole picture, irrespective of whether there's a known function of it or not. Okay, Madhya is asking, sir, can we use microarray data set for differential gene expression? Yes. RNA-seq is definitely better. Microarray is obsolete. So forget about the data that is there. You can utilize it for analysis. You can play around with it, but it's not as reliable as RNA-seq. So if you have to design a new experiment, go for RNA-seq. Microarrays are obsolete. I have done it. I know it. So that's why I'm telling you by first-hand experience. All right. So whole genome sequencing is about 3 billion bases sequencing. And most of it is having unknown function, but still we want to know, suppose we want to see the effect of the rest of the DNA, how is it changing, or if we just want to do it for characterization purpose, that we want to have the sequence of the entire tumor or cell, then we go for whole genome sequencing. But it is very expensive and it is very difficult to put together the entire thing. Again, just think about the ball of wool that you have. And if you cut it down into pieces and you have to put them all together, it's a much difficult task than if you're given a fragment of that wool and say, okay, take this one, cut it into pieces and put them together. So whole genome is like the entire ball of wool, and a portion of it is like whole exome sequencing. So I'm sure you would, all of you would say that whole exome sequencing is easier than the whole genome sequencing, and it is cheaper as well. But you miss the whole information. So that's the whole exome sequencing. And then there's also some targeted sequencing, because you don't want sometimes to go and see the entire exome or entire genome. Rather, you know that, okay, these genes are there, which we need to study. That is why we go for targeted sequencing. So in that, this is also a very lucrative work in the industries, that they are trying to develop panels. And I think I have a few slides on that as well. Panel of genes which represent a particular disease. So they have got panels for different types of cancer. They have got panels for different stages of cancer. They've got panels for different types of samples. So that is all through the targeted sequencing. The targeted sequencing is more like the Sanger sequencing, and but it is done in a high throughput manner. There is a comparative illustration of all these three. Whole genome sequencing, when we say reads, that means each fragment is sequenced 30 times. Reads is greater than 30x, because you're doing all 3 billion bases, so you can't go very deep. You can't do a lot of multiple sequencing with the same fragment. So it covers all genes and non-coding DNA as well, which could be structural DNA, which could be DNA with unknown function. Accuracy is low because again, you are dealing with a big stuff, and it will also have a turnaround time is high because you have to make a big library and you have to do the sequencing, and then it will also cost more because it is going to be more labor intensive, and it will be involving more sophisticated instrumentation as well. Thank you, Asad. Whereas in case of exome sequencing, as we see here, we are only looking at the portions of the entire genome which are known to function, which are known to have RNA. The exon 1, exon 2. So we are talking about the exon level. So these portions are sequenced, not the entire genome. These gray portions, they are not done. So now we are more focused, so we can increase the depth, which means we can sequence the same fragment from 50 to 100 times. And this is only 20 to 25 genes. So accuracy is better. When you've got a smaller task to do, you're better. Definitely turnaround time is there, but it's less than whole exome, whole genome sequencing. The cost that we are talking about, the whole genome and whole exome, now it's in the hundreds of dollars. So it's more cost effective. And this is the depth, again, 50 to 100x. That means we are able to sequence each fragment 50 to 100 times. And targeted gene panel is something that we know, okay, we need to know not all the 25,000 genes. We are doing only 10 to 500 genes. So we are more and more focused here, and we can do the depth, it could be up to 500x. So the accuracy is increasing, right? Because we are doing the same stuff multiple times here. So we are more confident about this sequence. The accuracy is pretty high. Rapid turnaround time is few days. Now people can do it in a week time, along with the analysis. Because after you generate all this data, there's a lot of time and work that is in putting these pieces together. This is the analysis part. This is the work of the computational biologist. And to tell you, they are the most highly paid because they are taking the data and they're putting them together and giving you the final information. So these are the three variants of next generation sequencing. So this was all about DNA, that the next generation sequencing is utilized for doing the sequencing of the DNA. So the first layer of the multiomics, DNA is covered through these approaches, right? So the second layer is the RNA. As I said, we'll be applying the same technique. Any questions so far? No. Okay, so we will move on. So RNA, the technique is the same. We are again sequencing every nucleotide there, and we are doing massively parallel sequencing. We call it as RNA-seq in general terms. This is somebody is talking about RNA-seq. It's transcriptomics, the second layer of the multiomics, and RNA sequencing, or in short, we call it as RNA-seq. Right, so this is the entire genome that is known, 3 billion bases. Now, from the genome, the RNA, or first the pre-mRNA is made. The one that I was telling you, that these are the dark red colors that you see here. These are the exons, and the light red color shown here are the introns. So the arrangement of these, suppose this is A, this is B, and this is C. So the arrangement of this could be changed depending on the need. So it could be A, B, C, or it could be B, C, A, or any combination. And this is just a simple 3 exon structure. If you look into some of my papers, there were 32 exons, 50 exons. So there are more exons, and that if there are more exons, it will create more complexity and more combinations. So once this is splicing is done, the intron splicing, the mature RNA is formed, which goes for the translation and making the protein. So RNA-seq is done for the entire RNA sequence, which is the pre-mRNA forming the mature mRNA, and then mature mRNA is digested to create the RNA fragments, and then it is converted into cDNA, that's shown here in the blue color, and then these are sequenced. The double standard cDNA fragments are actually sequenced. I haven't put that slide for QRT-PCR. QRT-PCR also employs the same thing, but it is a different chemistry altogether. Okay, I'll tell about it later on. So here again, you come up with the sequence, and this sequence will tell you what was the sequence of the RNA. Right? So with this, we know that the RNA abundance as well as RNA sequence. So mostly people have been interested in knowing the RNA abundance, that how much of the RNA is there. The differentiated gene expression has been extremely popular. If you look into the literature, people get excited. Oh, this gene is upregulated in this cancer. Let's knock it down and we cure the cancer. But again, the concept of multiomics tell them that no, don't get so excited. It's a bit early because you're not seeing the full picture. Right, then we have these fragments. We have to align it. This is all in silico work, so alignment needs to be done. Putting together the fragments of the wool that I just told you, that you have to put together those fragments to create the entire sequence, and then you can say whether it was splice variant A or splice variant B, because this sequence will be dependent on how this splicing was done here. So when you know the sequence, you will know whether it is BCA or ACB or ABC, whatever it was. So this sequence information will tell you which variant are we talking about. Then there is also a variation in case of RNA that you can do the whole transcriptome sequencing, which will be similar to the whole exome sequencing, roughly, because the whole exome is converting into RNA, and that's what the whole transcriptome would be like. And this will be known as whole transcriptome sequencing. So we take the mRNA, convert them into cDNA, and then you fragment them, make aliquots, and digest with the enzymes that are being used here. These are the TSP5091, and these are different enzymes which have got different cutting sequences. NIA3, MSP1, DPN2, and each company has got their kits available. They have got their own set of enzymes for making these fragments. And then once you have got the fragments, you can pool the aliquots and start to label them with the beads. And once you label them with the beads and then you sequence them after PCR amplification, and you can get the data about those fragments of the RNA. And again, here, Illumina is leading the industry by using its platforms, and you get good data. Here also, there's a multiple read. So you have got the depth, or you call it 50x or 100x or 200x. That's relevant for RNA as well. Now, we also have targeted gene panels, which means you are focused on a certain set of genes. If you look into the literature or if you look into the market, they have got multiple panels available. Some have got 50 genes, 100 genes, 200 genes, 300 genes. They will do it for you, and they will tell you out of that which gene is active or which gene is present or not present. This helps in the clinical decision making. So, but targeted gene panels is only useful when you know for sure that what you are looking for. If you know that, okay, I have to look for the status of the mismatch repair gene, say MSH2 or MSH6, you want to know the status because there's a cancer. This is that Asim was talking yesterday, hereditary non-polyposis colorectal cancer. We know that in that cancer, MLH1 is mutated. MLH1 does not work. So now with targeted gene panels, we just want to know about MLH1. So now we don't need to do the entire transcriptome. We don't need to do the entire exome. We want to know about the MLH1 so as to know whether this patient would be developing colorectal cancer or not. So there we go for targeted gene panels, where we are sure about a particular gene responsible for a particular type of phenotype. So it's more focused analysis. have to run a bit faster here. Instead of sequencing the entire genome panels, they focus on predefined set of genes, and they get you actionable insight like HER2 and Versta. They are also easily doable, so they are available in the common market. Examples here are HER2, EGFR, MET, and then there are cancer-specific panels, as I just told you. They're available. Here I found one gene panel. These are the genes. They were utilized 1, 2, 3, 4, 5, 6, 7. 7 genes were used to classify 9,716 human cancers, and they found 28 molecular subtypes. So this is the gene panel that they use for patient classification, that which patient belongs to which subtype, so that they can decide upon the therapy. But our target is personalized medicine, going for a single patient, not the. So if I were to say this, these will be 9,000. Yeah, here. So 9,716 human cancers, they were subtyped into 28 molecular subtypes. With the personalized medicine approach, there will be 9,716 molecular subtypes. This is what we are trying to achieve. And as I said earlier, we are confident that the development of technology will be able to do that. Okay, so single cell genomics is another variation where we are trying to understand every cell as an individual, and single cell genomics has also progressed with the help of microfluidics. We can isolate these cells, and then we can do the sequencing of the genome or transcriptome of a single cell. So this is not a single cell genomics. And there's another variation called spatial transcriptomics. This is also a very upcoming field where you know which type of cells are present in what location of the tumor. So like if you look at this particular tumor, there are healthy cells, there are mostly cancer. These are the ones that are on the way to become cancer. They are very close to be the cancer. This tells you which, and for the surgeons, you can guess which portion will they take out first. They will take out this red portion first if they know, right? So this is what is being utilized for the spatial transcriptomics. So this was a benign tissue, but when they did the spatial transcriptomics, they were able to find no. Because if you take the sample from here, you will not be getting the full picture. Or if you take the entire tissue, even then you will not be getting the full picture. But with the spatial transcriptomics, you know where is the malignant cells. So with that, we move on to the next layer, which is protein. I think I have to rush. A typical single experiment in protein is still not there. It's far, far behind than the nucleic acids, what we have achieved with next generation sequencing. Proteins are far behind because in a typical single experiment, we cannot do more than 8% of the human proteome from blood or 35% from cells and tissue. So we are still not 100%. In case of DNA and RNA, we can do 100%. And this employs the technologies like chromatography and electrophoresis. Electrophoresis it's pretty early. It's like Sanger sequencing of the DNA. 1D and 2-dimensional polyacrylamide gel electrophoresis using STS page or native page. Or it could be liquid chromatography using HPLC or other type of separation techniques. Then we have got the mass spectrometry, which is the current standard that we use for proteome analysis, the mass spectrometry. There's also shotgun proteomics, which is more like targeted sequencing. So shotgun proteomics is identifying a particular peptide and particular types of proteins and just sequencing them for the proteomic studies. There's also technologies called SciLac, iTrack, which are labeling the peptides and then sequencing them. So they use the radioactive techniques for labeling the peptides, and then they're able to sequence these. And then all the technology is also allowing label-free quantification because they use the time of flight, which is not here. So it's known as like Maldi-Toff. So they see how high the fragment flies, and based on that, they're able to characterize a particular fragment of the protein. So it's called label-free quantification. And this is complemented with the XRDs, X-ray crystallography studies about the three-dimensional structure of the proteins. NMR also gives you idea about the entire protein structure and how is it changing with any kind of mutation or anything. E2 hybrid tells you about the protein-protein interactions, that how these two proteins are talking to each other, because at the functional level, again, this is important to know. As we said in the DNA and RNA, we need to know the pathways. Here also, we need to know which protein is talking to whom and how, so that we can disrupt that or we can increase that interaction for a better therapy. And on the same line of the DNA microarrays, protein microarrays have also been developed and they have been utilized. They're still in use, but they are low resolution, high throughput screening of protein interactions. And so there are protein microarrays, and then you also have multidimensional protein identification technology, which is 3D level of identification of proteins. You can consider it as a spatial transcriptomics or a spatial proteomics, where you know which protein is getting expressed in what location. And since we are able to isolate a single cell, we can also do single cell proteomics. So single cell is nothing but isolating the cells from the bulk and then utilizing the transcriptomics or genomics or proteomics techniques from the individual cells. Here are some of the platforms available. This is Quantum C. It is for the single molecule protein sequencing platform, but the most popular is this one, Thermo Fisher's Orbitrap. It is able to analyze one sample in just 8 minutes, and it can identify up to 8,000 protein groups in a single run. But you know, there are more than 100,000 proteins that are available at a particular time, so this number is still very low. 15,000 proteins can be done if you allow more time, and this also works for single cells. There are many other variations available in the market, but this is the one that is very popular. Next, we go on to the next layer, which is metabolomics. So we have got DNA, RNA, protein, and now the metabolites. So as I told you earlier, the techniques are the same: mass spec, GC, LC-MS. We can also do the ionization techniques. So we modify the ionize these metabolites and then see their separation or their flight. And then we also have the NMR spectroscopy for detecting very sensitive changes in the metabolome. In case of metabolomics, you will often hear the terms untargeted metabolomics, which is again like whole genome sequencing. You're not looking for a particular metabolite, rather you are trying to understand the entire metabolites. And this is also done through the LC-MS. But you can do targeted metabolomics. It's like the panels of the genes that we talked about. It focuses on quantifying specific known metabolites. If you know which metabolites you want to study, then you have the panels for that, and you can do it again through LC-MS. So mostly, if you have got a lot of money, people go for untargeted metabolomics if they don't know what to look for. But if you know what the metabolites you want to study, then you go for targeted metabolomics. In my experience, we have a lot of computational tools. So we zero down upon which metabolites to look for by using the computational techniques, by using genome scale metabolic models, which can be done with the help of the RNA-seq data. So we don't spend a lot of money in doing the untargeted metabolomics. But yes, some groups might be interested in doing that as well. There are other omics, which is this list is also not complete. There are many more omics. If you look into the literature, you will find omics next to everything. So lipidomics, about the lipids, glycomics or glycoproteomics, because proteins, they get post-translational modifications. So this could be about the sugars. Then you have got the microbiomics, about the microbiome. You can use the metagenomics as well as metatranscriptomics. Interactomics, how these are interacting, the biological molecules like protein-protein interactions. If you do it in a high throughput level, you call it the interactomics. Radiomics, if you are doing a lot of imaging, then you call it the radiomics. Fluxomics is another step. You are measuring the flow of these metabolites in and out of the cell. And if you're doing it at the high throughput level in a global perspective, we call it fluxomics. And single cell omics, I have already talked about. When you're talking about a single cell, you call it a single cell omics. Now, the most important part, and I have a few minutes for that, I would request Professor Hamid Ashraf to give me 5 more minutes, who's the next speaker, because all this is going to create a data deluge. A lot of data will come out. Now, if you are not smart enough to handle this data, this will be of no use. We need to generate information out of this data. And for that, we need to be able to analyze single omics. We need to be able to integrate them, and then we need to make the clinical sense out of it, right? And then we can use it for either disease subtyping or patient stratification. And don't forget, we have to look for a single patient. So this has to be done at the level of single patient one day. We can also use it for biomarker discovery that I'll tell you in my subsequent lectures that we discovered some biomarkers, we discovered some targets using these approaches. And we also can understand the regulatory mechanisms of the function of these genes. And ultimately, this can lead to drug discovery and development as well. Once we know the target, definitely we can know the drug that is going to act against it. So there are different strategies. We can do the integration. Integration means the integration of the genomics, transcriptomics, proteomics, metabolomics, epigenomics, how to put them together, the jigsaw puzzle. So this can be done at an early integration stage. So when you generate the data, you can start integrating at that stage, or you can even filter the data. You can clean the data and then do it in the later stages. So that's why we have got early, late, intermediate, or it could be hierarchical, which means that first we'll do DNA, then we'll do RNA, then we'll do protein. So this is hierarchical integration, which is more like a funnel shape. You start with a lot of data, and then you start zeroing down upon a particular type of proteins. And there are several computational methods used, like regression analysis, lasso, elastic net. Bayesian methods are quite popular. Neural networks are quite popular. And these days, even multimodal systems are available for integration. I think I gave you some examples that Microsoft is working on, Google is working on, because these guys they're already very smart with the data. So now they're coming into this multiomics arena, and they're trying to utilize their tools to analyze these multiomics features. But the challenges are there. The data is very heterogeneous, coming from different patients, different level, different stages of the disease, different cells. So this heterogeneity needs to be taken care of. It's like you are being asked to analyze a population of people living in a particular area and asking for what is their pattern. It's very difficult to see people moving in and out, and people having different habits, people having different shape and sizes, skin color, and so there's so many heterogeneity. It's very difficult to tell the behavior of the people from that. But yes, now we can do that, at least for the multiomics. High dimensionality, each omics is a dimension. So genomics is a dimension, proteomics is a dimension, transcriptomics is a dimension. So we need to make sense out of these dimensions and give a target that can be worked upon, and the drugs can be developed, and it can be used for therapeutic purposes. Batch effects, which often happens in labs, that you do an experiment today, you do it tomorrow, it's not the same. This is called batch effect. So it should be taken care of that even one person does it and the second person tries to do it, they cannot do it. So these are batch effects, which is also creating problem for the data filtering and data analysis. And most importantly, data interpretation. Clinicians have to be trained for this. We as molecular biologists, biotechnologists, we have to make it as easy as possible for people to understand. And computational demand, which means you need a lot of infrastructure, servers, and the computational tools, the high-speed internet and high-speed processing that is needed for that as well. And this is my final slide, and this is in my this is a publication that we did recently, multiomics for the cellular plasticity. I'm not sure if it is having a good resolution, but it's the same concept. In everything that I just told you, it's in one slide. That we start from a normal cell, we try to isolate the cell, we do the data generation, data storage is there, then we analyze it based on the genome, epigenome, transcriptome, proteome, and metabolomic sequencing and analysis techniques. With the use of artificial intelligence and other tools, computational tools, we can go for biomarker discovery, identifying therapeutic targets, mechanisms and drug resistance and personalized patient profile. And this is what my dream is, to one day come up with something that can be given to every patient in a personalized manner so that we can save lives. And the ultimate objective is to save lives. If I'm able to do it for one cancer patient, I'll consider my career fulfilling. With that, I'll say thank you, and I'll take some questions if there are. Otherwise, I'll hand it over to Dr. Hamid Ashra. Okay. Yusra Jamal, I think you should check your, if you were there in the yesterday's lecture, we have already posted the drive link, so you can take it from your colleagues. Safa Zafar, if a researcher wants to reduce cost and data volume while looking for specific mutations in a known cancer gene, would they be more likely to use whole transcriptome sequence or obviously target gene panel? Because when you want to do more sequencing, you increase the cost, you increase the data volume, you increase the complexity. You definitely go for a targeted gene panel. But again, you're missing some of the pictures. For discovery aspects, yes, you should go for whole exome, whole transcriptome, whole genome. But for the actionable items, you will need to go for these targeted gene panels. But Malati, what is the difference between exome sequencing and transcriptome sequencing? Exome sequencing is for DNA, transcriptome sequencing is for RNA. That's a simple, straight answer. Exome is for the DNA, and transcriptome is for the RNA. Right? But yes, exome is that portion of the DNA which is making RNA. So if you do the transcriptome sequencing and exome sequencing, you might find similar results. But still, exome is you're taking only those portions of the DNA which are expressing genes. Whereas in case of the transcriptome, you are seeing the alternative splicing as well. The exons might be rearranged, so you would find different splice variants. But in case of exome, you will not find splice variants. All right. If that answers your question, in that case, when should we prefer exome sequencing? If you want to know about the gene expression, you go for transcriptome. If you want to know about the mutations or copy number changes in the genes that are being expressed, you should go for exome sequencing. I would like to say your work seems very insightful and promising. I would like to ask, what aspects are you trying to cover using AI in your work, and how will it shape future research approaches? Okay, Sandeepan, just wait for my subsequent lectures. You'll get to know more about what we have done and what we are going to do. Okay, so if you are not satisfied with my answers, you can send them here and I'll try to type it for you, okay, while the other lectures are going on. Or you